Okay, so the problem is that chemistry is highly dependent on the environment, and this is hard. There is another important question. We don't have other examples of life. We only have our own model. And we know of no life beyond the Earth. And actually, it doesn't seem to be easy to detect it out of the Earth. This work, actually led by our friend Armando here, they realized that actually right now there are detection limits in the rovers and in the systems that we are sending now to Mars. They are not even, some of them are not able to detect some types of life here. So probably they're in the limits of what we need. So we have to develop technically and develop formally all this line of research, which is quite promising. And always we shouldn't forget that beyond a biologist or an engineer who is really happy trying to detect what could be a fossil or a geologic, if you want, a print of a biological stuff, you have a geologist reminding you of the fact that geology, without the need of biology, is able to create fascinating complex structures. So be careful with that. All right, not only that, the main problem for us, for the people who do models, unfortunately, current biology cannot help us too much. It is far, far, far too developed. Right now, in the cell, really fast, we can replicate RNA, DNA, has lots of very, very complex metabolic pathways. None of that was in the beginning of life. So we have to invent something completely different. A good point, according to Cleves, as most of you know who he is that the origin of life probably used much simpler, that's clear, much more inefficient, and much slower processes. But, however, the good point is that we had a lot of time, 100, 200, 300 million years, and we had a huge world where we could try many and many parallel attempts, and at the same time, there was not already another life that would try to eat anything it created. So we have a lifeless world, we have many parallel attempts, and we have a lot of time, and that's good. Okay, so what do we propose? And of course, there are many other people, also in chemistry and also in biology, systems chemistry, system biology, and complexity. What we propose is that complex systems are composed of many agents, and what happened in the beginning of life was one of these complex systems interacting. We need things to interact. What is a complex system? In a complex system, there are emergent properties that appear in the sense that you wouldn't see them if you only look at each of the agents by itself. And the origin of a cell probably was this kind of interacting agents, and perhaps life is in fact an emerging property. This is what we want to see. Right now also we have something very important. The development in the last 10 years, 20 years of computational science and data science. And so we have now complexity science, artificial intelligence, statistics, all this together with very, very powerful computers makes this moment, this decade, I think the critical one to see whether complexity can really shed some light or cast some light, important light on the origin of life, or the problem is too difficult and we have to go on with different approaches. All right, who are we and what do we do? This is a group. Carla, Marina, and Marcos, these are my students. We are the Complexity Astrobiology group. We belong to the group of periodic chemistry and physics of complex systems here at CAB. And our main objective is to create a bridge, as Rita was saying in the presentation, between astrobiology and complexity. Our tools, we are not experimentalists, okay? We are very happy that all the people around do fantastic experiments and we get the data and we can discuss with them, but I wasn't born for doing experiments. I can't even change a plug. So what do we do? Complex networks, mathematical and numerical modeling, data science, Bayesian statistics, artificial intelligence, those are our tools, okay? And specifically, what do? We're trying to understand the increase of complexity from the very beginning, from the creation of the first molecules in outer space to the current biology in this nice complexity ladder that Marina created a few months ago, and I've put it in like I don't know, 20 talks so far. I really like it very much because it explains really, really well what we're doing. In this little Excel, if you want, structure, I'm going to show you the different lines of research that we're doing here and how network science can give a lot of information here. In this first column, we have, when we study a system, what is an agent? What is the agent here? The agent can be the most simple one. It can be an atom, can be a simple molecule, can be a polymer, okay? Whenever they interact, they can be a molecule made of atoms, it can be a chemical network made of molecules, and could be monomers, could be organisms, it depends on the case. And here we have these two lines of research related to astrochemistry, which obviously happens in the interstellar medium before the solar system was created. This is the periodic chemistry, and this is the biochemistry or biology right now. In the first step, we've been working for the last 3 years in the emergence of complexity in the interstellar medium. What did we do here? I created with a student this a crazy computational environment called Networld, in which it's completely abstract, in which you give different nodes to it, like 20 nodes, 20 agents. Let it interact. They start to interact, they grow, they get divided, and they create different structures, which are in fact some kind of, it's like a network chemistry. So something very abstract. But when I got here, I met many people, among them Izaskun Jimenez Serra, who is a collaborator of this work. And he said, Jacobo, what you've created, in fact, can be applied very nicely to understand how the first molecules in the outer space, in the molecular clouds, were created. And surprisingly, at least for me, not only qualitatively, but quantitatively, we could predict the abundances of these real molecules just by a system that has nothing to do with real chemistry. It was very, very promising. One nice point of this work is this. Here, this is our data obtained from my model that has nothing to do with reality. And we saw that the abundance of each of the structures that we create correlated quite nicely with the number of paths, with the number of different paths that created that structure. That is, imagine we're talking about my system creates, I don't know, like a network that is a square. To get a square, you can organize it, you can create it by different ways. If you have 4 different nodes, you can have one pair, then another pair, and these 2 pairs get together, and so you have a network. So you have a node, a square. But also, you can have like 2 squares together, and they get divided, and they create 2 different squares. So we said, if in my model it has nothing to do with real chemistry, we've seen that the things that are more abundant are the ones that can be created in more different ways. Does this happen in the universe? Is it true that in the universe, things that can be created in more different ways are also more abundant? And surprisingly, we looked at real data of different different molecules in 4, in this case 2, in 4 different molecular clouds. And we found that there is, of course, with a lot of noise, a very nice, very, very nice correlation in which the more reactions potentially can give rise to water or to CO2 or to NH3 or whatever, the more reactions that creates you correlate with the real abundance of this. So this is the paper. Right now, what are we doing? We are doing a step forward in which we apply artificial intelligence, which we are starting with this, in which the idea is to train my system, this network, in order to be able, if we can get information of the abundance of real molecules just by looking at their structure. It would be very nice in the sense that it would give us potential targets to look for. Because, I mean, we have already detected more than, well, astrochemists have already detected more than 300 molecules in these molecular clouds. But some of them, they have to decide what to look for. this could be nice to get some targets. Okay, this molecule, you haven't found it, but it seems that it should be very abundant. Attack that. So this is one of the nicest lines that we're working on. Another one. Here, another way of studying, much more connected to reality, if you want, of studying the chemical evolution of the interstellar medium. To get to study this, if you want, chemical reactions of different type. In this case, my student Marina, with the aid of Izaskun, Jimena Serra, what she did is to study the phosphorus chemical reactions, okay, and the chemical network, if you want. And she faced the problem of what we call the interstellar phosphorus problem, which is that all models so far show that PN should be more abundant than PO, but all observations clearly explain that PO should be more abundant than PN. So we did a model, we studied it formally, we were able to explain it, actually to solve it analytically, mathematically, which makes us understand it much better than it had been understood so far. And at the end, we applied Bayesian inference, which is very useful in this case to mix real data with a model in order to constrain the real values of the most important reaction rate coefficients. For example, we found out that for this reaction here, which is quite important, nitrogen plus PO gives rise to PN plus O. This line here is the value that the current astrochemists are using, and our prediction is that it should be 50 or 100 times smaller. So this is a very nice way of saying, hey, actually more than 90% of the values of these parameters that have been used in astrochemical models are not known. So this way we can see which are the important ones, which are not so important, and which ones should definitely be recalculated with perhaps experimental or computational ways, because they're clearly far away from what they should be. So this is also a very nice work. All right. Now we leave the interstellar space, we get into the Earth, in the beginning of life. This work is done in collaboration by Carla, my student Carla, and in collaboration with Carlos Briones, that you all know, he's down there. So where is the idea here? The idea is, let's attack the origin of information, one of the main columns of the origin of life. How the RNA, the first RNA, polymerized and replicated. It's clear that, as I said before, the modern way of doing it is not going to help us. It's extremely complex. You need a structure that is much more complex than the real RNA to make it replicate. It's like right now, where we construct cars using robots, but it's clear that the first car was not used, it was created by a robot that is much more complex than a car. This is more or less the same problem. So what we're doing here is to develop a model in which we quantify the influence of the environment, whatever you want, whatever you understand by anything that you understand by environment, pH, salinity, temperature, whatever, in how the RNA was first polymerized and then replicated in a clay surface. Which actually, in the group of Carlos, they are doing experiments in this sense, and it's a very long experimental. For that 30, 40 years, it's been working. People have been working on this and how the RNA can polymerize on clays. And in this case, what we do is it's only numerical and mathematical. So it's only here there are experiments. We are seeing how this each color is a different nucleotide, how the RNA can polymerize. Other nucleotides can get stuck by hydrogen bonds and this kind of relationships, G with C, A with U, G with U, as you all know. And then you get this copy, which is the copy of the original, can get into the pool again. It can find another surface here, in this case, where it can get stuck. It gets copied again, and the copy of the copy is a more or less efficient copy of the original. This is the main idea, of course. And this is what we're doing with lots of computers at the same time. It's very, very computational consuming, but we can try lots of different environments, lots of different things. For example, we're very excited with the idea of the genetic alphabet, because in our model, we can work with an alphabet of 4 letters, of 2 letters, of 6 letters, whatever you want. And what we've seen is that the 4-letter alphabet seems to be more efficient than the rest, a little more, but especially if you accept the RNA as it is today, where you can have G and C, one pair, A and U, but also G and U, this last one, even if it's not so stable, this makes it much more diverse, and it creates a lot of diversity that is like 1 order of magnitude more powerful than the rest of alphabets. So this was not the idea of the work. The idea of the work is to create like an environment to see how RNA could polymerize and replicate, but still, now we have our toy and we can try things. And so it seems that it's giving us nice information about whether in this fight in the beginning, whether why 2 or 4 or 6 letters in the alphabet, but nobody knows really why these 4 letters was fixed. Well, this could give us some information on that. All right. We forget information and we get into the other big, if you want, big column of the origin of life, metabolism. In this case, Marina, with the help of Marta Ruiz, which I don't know if she's here. She's not, she should. Perhaps it's, we are working together with Gabriel Piedrafita and trying to make a very, very simple model of if you want, like a proto-metabolism in which different molecules can stick to other ones and create like polymerase, but also they can get divided. And so we have like a small reaction network that can be studied, especially from the point of view of autocatalysis, to see whether these small molecules can interact, and they can interact, they can get divided, they can grow, and they can make the cell grow, which would be very nice, because in some sense, the growth rate is like a proxy for fitness. Because if the cell grows faster, it will get divided sooner, and then we can suppose that that cell will, at least at the end, push the other type of cells and be the one that survives. So this will be like a model of the origin of metabolism, but always from our point of view. We don't really have real molecules, they're just agents. Everything is very simple. But as they are very simple, we can do much more work and much more diverse than with other more reductionist points of view. One detail, all the work done so far was based on, okay, whether from this point of view, if you have an autocatalytic network, then things will work. No, that's not true. Whenever you talk to people, to real chemists, they tell you the most important stuff is that your cell has to be stable and has to be kinetically reasonable, okay? So this is very, very, very important. It's not enough that you can duplicate something. You have to duplicate it for a long time, and it has to be stable. And so we're working on all those kind of points, all those points of view of how the different kinetics depends on the parameters, and how can we be sure that the kinetics work. And now we jump to this last line. We reach the modern world, okay? And I'm going to explain you with more detail, because I think it's quite pedagogical, as I said, our last work that has to do with the interaction between the protein network of a virus and the protein network of a cell at different moments of the infection. You will see that, surprisingly, the tools are just the same as we are using for the other works before. So this is a good thing of complexity. You can go from one subject to the other, and of course, you must always work with people who really know about that part of the work, but you can try to collaborate, and this institute is perfect for that, as you all know. And this work is being done, it was done with Raúl Guantes from the Autónoma University. So what have we done here? We've studied 4 viruses, 3 herpes ones, and a pox virus, the vaccinia. If you ask me, that you will, I know, why didn't you use RNA viruses, which are the ones that are more important now for obvious reasons? Because they are too simple. They have very, very few proteins. And so you cannot talk about the interaction between networks. If you have a network of several thousands of proteins, as it is the network of the human proteins, and a network of, I don't know, 7, 8, 20. They're too small, okay? So we chose this 4. And what we're doing is to understand the viral infection as a dynamic process, in the sense that host and virus proteins change at different times during the course of the infection. We went to several papers and we took the information. So we didn't do the experiments, but they are all based. All this is based on real data of the proteome, of how many proteins have been, if you want, created at different moments of the infection of the cell by a virus. Okay, after 25 hours, 50 hours, whatever. So this is the idea, and we want to create networks, because I feel comfortable when I see networks. Okay, this is my tool of work. So what do? Each node is going to be a protein. Okay, get this idea. Otherwise, you will not understand anything. Each node is a protein, either of the virus or of the cell. And but not anyone. We're not getting this 7,000 or 10,000 proteins. No, we're only focusing on those that are significantly altered with respect to the non-infected control case. The other ones, we suppose, that are not important in this case. Second one, how do we connect them? Because every network has two things: nodes and links. Like the metro, for example, in Madrid, no? Have stops and you have connections. What do we do? How do we do it? Link proteins. What we do is to link proteins with high similarity or anti-similarity in expression profile. That is, this is critical. So listen here. This is the number of proteins, okay, created at different times, measured at different times of an infection. And these are always, this case, I think it is, yeah, one of these is of the virus and two of them of the host. These two are very similar, as you can see. And this one is very similar, but it's the opposite. This is a first idea. We suppose that two proteins are interconnected if their activity is very similar. You can say, hey, man, this can be true. It can be true sometimes, and sometimes will not be true. Two proteins that have nothing to do could have the same activation pattern. It's true. But we'll see that at the end that the results fix and everything goes quite well and agree. So we suppose that if you do it with a little bit of care, because all this part is quite annoying of deciding which proteins you should choose and which not. If you do it at the end, there will be very few that shouldn't be there, and most of them work. So a node is a protein, and two proteins are connected if and only if their activation pattern is very similar, both positively and negatively. Okay. And now we do this for these four cases, and we get these 4 nice network of connections of these 4 viruses. Okay. This is an example. You can see it bigger here. What do we do now? By the way, these are the virus graph. So all these are the proteins of the virus, and all these are the proteins of the cell. Okay, and these are the connections. Next step, in order you to understand what I'm doing here, I want to divide this structure in different parts. So I get an algorithm called Leiden. There are lots of them. And I tell this algorithm, please divide the network in the most logical structures, okay? And it gives me one structure here, another structure here, another here, and another here. And here is the virus. If you paint it, you get this strange structure, okay? But it's very clear. This is the virus here, okay? There is a very small network here, which is this one here, very small. And then you have these two and these two. And so now we have the networks. And we have the network with the connections, and we have the different structures. And this is what we're going to work with. And this is very nice, because we get to the first result of this part of the work. Protein expression networks are a network of networks. Can be understood as different networks connected through a little number of links. And I love this stuff, because I've been working with that for a long time. Actually, 10 years ago, we published this paper where we studied, or we realized that we could formally understand the interaction between different networks when they interact. And we showed that there are some strategies that are good for small networks, good for big networks, depending on how you connect to them. This could be applied to economy, for example, when you want to join companies, but also to ecology, can be applied to lots of subjects. Anyone in which you have different networks that are going to compete or to collaborate. And so we have a whole formal structure already created that I call the theory of interacting networks that we can apply here. And this is what we did. So we have like a whole formal stuff, whole mathematics that we can apply here. So I feel comfortable with that. So now the next step, you have to remember something. We are studying a system that moves with time, evolves with time. But even if it's evolutionary, we only have one network. In one network, everything is together. So there's no dynamics there. Okay, this is the problem with this. We didn't want that the links change it only because every connection means that all your whole evolution is very similar. So the whole infection is plotted in only one structure. So how can we study dynamics here? This is the idea. We get, in this case, for two proteins, and we study their abundance. In the moment that the abundance becomes twice the one that had in the control, in this moment, we decide that it has been activated. Okay, this one would be activated after 5 hours, and this one after 18 hours. And we call that the response time. So each protein is going to be activated at a different moment. And now we have a dynamics, have something that we can measure. If we paint the distance to the immediate early viral genes, which means these are the viruses that start the infection. Okay, they are you can go to a data set and you see them. For all these 4 diseases, we know which are the viruses that start triggering the system. Okay, over this network, we can measure the distance, and the distance is just the number of steps that you get from this original virus to each protein. You can measure that, it's very easy. And if we put this distance against the response time, what we find here, and here you can see it through for each community, we can see that there is a clear relationship, which is quite nice, between the moment in which you are activated and the distance to this point. This viral genes will be around here. This is the virus. This is the first. In this case, this is the vaccinia. The virus here has 2 different communities, 1 and 2. Look at this. They get activated in the beginning. After a while, after 5 hours, this small part here gets activated. After 10 hours, the red part is activated. And after 15, 20 hours, you get all this part activated. So what do we have here? We have a nice activation wave, in which even if we are starting with a network that does not evolve on time, we can understand, we can see how the virus is able to activate different parts. First, it activates at the beginning. This will be the 2 ones that activate first. But then from here, you have like a wave of activation of this part, and of that part. So there is some kind of dynamics there. Another question. Okay, you're talking about movement of some kind of activation dynamics, but you haven't told me about, well, you also told me about different communities that interact, but what are those communities really? Are they just a mathematical formal structure, or do they have some biological meaning? This is the key question. Is there a function related to that? And so we decided to attack this problem, the problem of which is there any biological relevance for each community? Because that is what really virologists want to hear. All this mathematical stuff obviously is not very useful. And so a couple of questions, just for you to understand what we're going to do. In the world of network science, centrality means importance. Okay, if you want to measure the importance of a node in a network, you call that centrality. And there are different types of centrality. Here we're going to work with 2. The most basic one, degree. The degree is the number of neighbors of a node. In Facebook, your degree is the number of friends you have, okay? It's the easiest way of measuring the importance of a node. It's completely local. It has a problem. Perhaps your friends are not very important because you're not very important either. But if you want to understand how, if a node is really dynamically important, you can have another quantity, which is in this case the eigenvector centrality. The eigenvector centrality of a node gives you the importance relating to your friends. If you have very little friends, but they're really, really good, yeah, I don't know, if I don't know, the Minister of Science is your cousin, then you will have a lot of information that none of us has, even if it's your only friend. So both quantities are important. If you have a lot of friends and even they're very important, then you control the system. So these two quantities are very important. By the way, the eigenvector centrality, I'm very fan of this quantity, but not only me, is the one that Google uses when you write, for example, I don't know, Ramon y Cajal in a web page in Google, and you find this ranking of the most important pages. This rank is done by this system, or very similar to that. What none of us know, the secret of the Coca-Cola of Google, is how they can measure this quantity of such a huge network. This, I mean, any computer can do this in really, really fast time for, I don't know, 1,000, 100,000, 1 million nodes. But Google has millions of millions, I don't know, a huge number of nodes. I don't know how many. And they can do it really fast. This is the key point, but the mathematics is very easy. So we have these two quantities, degree, that tells you if you are locally important, and eigenvector centrality, which tells you if you are globally important. By the way, for any people who like mathematics, eigenvector centrality is calculated as the eigenvector associated to the largest eigenvalue of the matrix that represents the network. This is a very, very easy calculation that any computer does with just doing like this, if it's not very big, okay? So, all right, let's get the case of the Epstein-Barr, you know, this virus that we all have, probably, and we will die with it. And in this case, we put these are all the proteins of the system with its degree against its eigenvector centrality, all right? These are the virus proteins, and all these ones here are the host proteins. So the first result, host and viral subgraphs show very large differences, implying different dynamical behaviors. I mean, this is not so strange. What does the virus want to do? The virus wants to wants to control the proteins of the cell. And the protein of the host, what are they trying? They're trying to defend themselves. So it's clear that they have different strategies, which is clearly here, okay? Virus here, host here. Different colors are different communities, by the way. But now we decided to look at these nice tongues here, and we said, let's see what they are. And it seems that we call them leader proteins. If you get the proteins that have more eigenvector centrality and more degree of different communities, it's quite nice because they see that they have some functions strongly, if you want, strongly marked, strongly over average, okay? So it's really most of the nodes that are in each of these little boxes have some functions overrepresented. So you can really say that, and actually not only that, some of them are known to be very, very important already for the infection. So this is a nice way of, it has resulted to be a nice way to see which are the proteins that really are moving the situation. So two ideas, as I said, different dynamical behavior of the virus and of the cell. And the second one, also important, the leader proteins are associated to specific biological functions. Some of them, some of these strategies are typical of the virus in itself, but some of them are general. It depends. But we did a very, very, we are not virologists because this is a methodological work, but we did some kind of study, of biological study of all the proteins. And it's clear that right now, the next step would be for virologists to see this list of proteins and see whether they all knew they were reported to be important, or perhaps it could help us again to find targets of new proteins that nobody had looked at and perhaps could be useful to understand how this infection works. Regarding astrobiology and the whole talk that I gave you right now, so far, systemic work, what we present here, I really defend that. I think that could boost the discipline, complementing all the work that has been done for many of you and for many other groups in the last 50 years, but from this more interdisciplinary point of view. And because, I mean, new approaches are always welcome, especially for the questions that have been open so long and seem to be so hard to explain, like, for example, why are we here? This is all. Thanks a lot. And by the way, people have work here. I almost forgot. I would have finished with a lot of haters. These are the people who have worked in this work. Okay, my colleagues here, Marta Itaskun, Carlos, Raúl in the Autónoma, Fernando in Sweden, Adrián is now in Graz, and all the students. Thanks a lot.
Thank you so much, Jacobo. We are now taking some questions here.
Hello, Jacobo. Thank you for that talk. I am very impressed. And the question I'm going to ask probably I need to think about it for a week, but I have to ask here, and it's probably not well thought, but if you go 2 slides back to the eigenvector centrality that you just explained. Yeah, so I understand that it makes sense to me if we translate this to the biological perspective. It makes sense to me that the viral proteins and the host proteins are separated because they have different targeted functions and everything. But I do not understand why there is more eigenvector centrality in the viral proteins in respect to the host proteins. Is it perhaps because, based on the definition you've given there, is it perhaps because the viral proteins have to be, it's obligatory that viral proteins have to be very related to host proteins to exert their functions? Or
actually, well, all questions are very good, but this one is especially interesting because it is true what you're saying. In this case, this is completely, well, I will explain the question in case you didn't get it. He's telling us that all the virus here are over all these parts, okay? This is general. This is not general at all. Of the 4 viruses, this is the only one that has the virus over it. And the point is that I didn't explain the details, but in order to have a lot of eigenvector centrality, you have to be very, very well synchronized. If you have a small network, but it is very, very well connected, and it's connected to the other one through very little links, you might have, it's not very low, it's not very useful, but you can have a lot of eigenvector centrality. So in this sense, I wouldn't be worried for that, because it's just the case, The important thing is that they're separated. And in this case, it's true that in the Epstein bar, you can look at here. This is not, let me show you, look at this. They are not many, but, oh, sorry, it's here. They are so well connected, so synchronized, that gives them a lot of dynamical importance. But in the rest, they are not so, I don't have here, perhaps I have the pictures of the rest, I don't know, I think I don't. But you will see that my work here. I think somewhere I have more of these pictures. Oh, sorry. Wow. No. Here. This is just, there. You have here another one. In this case, it's the other way around. So it doesn't mean much that it's over it, okay? But you will see the same structure.
And just to a quick follow-up, sorry to retain the microphone, the fact that they are so clustered, so close together in the virus, just to make sure I understand it well, means that the proteins are all acting towards one goal. Their functions are similar, and thus their activation rates and activation curves are similar.
A virologist should say that. If it is because of the function, what we see is that their activation regime is very, very, very similar. But perhaps each of them could be doing different things. Yes. We don't really know.
Okay, thank you very much.
Any more questions?
Thank you so much. Very visual summary. Thank you, Jacobo. So I have also a question that may be wrongly posed. Please help me explain it. When you show these beautiful graphs of networks, in this case for the viral infection, but I assume in everything you could have something like this. Are there networks that are more robust with respect to failure, and are there networks where you can target a node and say by cutting this node, the system is going to fail?
Definitely. This has been studied for the last 50 years. Actually, one of the most important realistic systems that pushed the science in the 60s, it was some blackouts suffered in US because they didn't know why. Perhaps there was like a small power grid place in New Jersey. For some reason, it didn't work. And for 1 week, 50 million people didn't have light. And so they realized that robustness is very, very, very important in network science. So there's a whole discipline in network science studying robustness.
So then, as a follow-up, bringing this to the viral infections, let's say you have this viral network expansion map. Can you then design a target place to attack to, so that by breaking that point, that communication, that protein expression, the infection dies?
I would say that is a bit too ambitious.
Okay.
In the sense that we can't forget, it would be useful to find which are the ones that you have to study.
Mm-hmm.
But from the point of view of networks, of network science, we can't forget that what I'm showing you is a bit tricky in the sense that two nodes are connected if their activation pattern is very, very similar. But you should first study whether there is a real interconnection between them. If there's a real interconnection, then that would be a good way. Okay. But it definitely has to be different steps to make sure that what you're doing is correct. It's not just like saying what I say, I understand with you. Otherwise, we could say, hey, let's kill all these proteins in the middle, and this will not work. I'm sure that the virus would find other ways to do it. Okay. Perhaps Carlos can tell us a little about that.
I think this is a very good observation, MP, because in general, you select the targets depending on their function, not the connectivity. So this could be an alternative approach and could be very, very interesting. Why not? And in that sense, I would like to ask you, you have selected different viruses, quite different for this last part of your talk, very nice talk, by the way. And what if you select evolutionary related viruses? I mean, belonging to the same family, for instance. Could you perhaps see a movement through the network and being able to detect some key points in the evolution of such a family? For instance, a new protein that starts a new sub network inside the whole network or something like that?
I suppose that if they belong to the same family, their network should be quite similar too. So I guess we could compare them. The truth is that we have selected these four because they were, it's like when they asked Hillary why he wanted to get up the Everest, and he said, I want to go there because it's there. In this case, we chose these four because they were the ones that had the most detailed information at different moments. We're doing a correlation with 5 or 6 points because they only measured the infection at 0 hours, 10, 20, 30, 40, something like that. So we were obliged to get the data that would fit better to do this. If those data are available, it would be very nice. Instead of getting such different networks, to get very similar and see whether evolution has shaped them. It would be nice, yeah, but we need the data.
I think that could be very interesting, for instance, nowadays regarding influenza viruses and trying to.
But we have again the problem
how they jump from a host to another, perhaps.
But they have a very low number of proteins.
Yep. So
the structure wouldn't be so good. We did it with I think HIV, and we also tried SARS-CoV, but they have, I don't know, 20, 30 different proteins. So that's not really a network. it's not so easy to get information from there.
Maybe if you could consider some motifs inside different proteins, for instance, hemagglutinin and so on in this case, or even regulatory elements in the genomic RNA, which also play a role in the interaction of the whole system. If you could extend, you could perhaps apply the same technology, let's say, to the study of RNA viruses, and could be very, very interesting.
Definitely, the next step should be to contact with people who know about viruses, virologists. But from this point of view, from this point of the work, the next step should be applied. People like you or whoever who say, hey, I want you to look for this stuff. And then we can focus there. Because the mechanism works. But of course, I mean, at the end, we explain something of the functions, but it's clearly methodological work. And so the next step should be to apply it.
Thank you, Jacobo.
More
Any
questions?
more questions
Doubts?
here? We still have a few minutes. Well, I have a very basic question, probably. So at the beginning of your presentation, you were talking about modeling, prebiotic evolution, and, so RNA replication, and you talked about physical conditions that you can model all of that. So I was wondering, what would be those ideal conditions, according to your models, for a proteome metabolism or for RNA replication or anything that you have studied?
In fact, what we have found in the work in which we are studying how the RNA polymerizes and then replicates, we've seen that we definitely need a fluctuating environment. There is no way we can replicate RNA if the environment doesn't fluctuate. When I talk about fluctuations, I'm talking about pH, perhaps, salinity. Imagine that there is like some water with the tides. You get some water in a place, and then this water starts to evaporate. So salinity grows, and then the water comes again. You could talk about temperature, night and day, or perhaps longer, regarding summer and the winter. There are fluctuations of everything, and we've seen that actually there are like especially good periods, so optimal periods for that. And now the problem is that we are trying to discuss with Carlos, with Carla, and with Adrian, how to connect this kind of abstract quantities with reality. Okay, we have some periods which are really, really nice for the replication of RNA, but these are periods, computer periods. Can we connect this with real time? So we're working on that.
Sorry, what, competing periods?
No, in the sense that if you have an oscillation, essentially we have a period. It's not the same.
Okay,
A tide than a day, than hours, than, for example, the waves, which are really fast. But of course, we can say things qualitatively because the model is very simple. But it's hard to say whether we need things that happen, that change with a period of months, which could be probably the temperature. It's hard to say that. So everything is qualitative.
So we need gradients, but do you have any particular?
No, not gradients, not gradients. We need fluctuations, which is not the same.
Sorry.
Same.
But do you have any parameters that you find that are better, or these are just, they do not have a physical, like a temperature?
This is what I was trying to, I didn't explain it very well. What I meant is that we have an environment that oscillates, but this is what we are just changing is the probability of these structures to get stuck and to get divided. And this can be done because of temperature, salinity, pH. So we have only one parameter that englobes all the environment. So the problem is that, of course, we have a very simple model, and we cannot say, hey, it's pH. No, it's temperature. No, we cannot do that. But the main point is that fluctuating environments are necessary. And actually, this has been done, it has been shown in the last years, 5, 6 years, in all experiments. So it seems that both experiments and theory are colliding. And in fact, I only explained the numerical stuff, but we have also done mathematical work, and we've been able to mathematically prove that oscillations are necessary to create an RNA replication, which I think it's a nice work.
Thank you. So Any other questions here? Yeah, we have.
Hello, nice presentation. I like the question of Rita because I was thinking of, have you tried like wet dry cycling, which would like affect all these parameters, salinity, probably pH, and of course it will concentrate all the molecules. So probably it will make more easy for them to polymerize or interact.
Again, I have to answer more or less the same. All the parameters, I mean, all this information, wet, dry, temperature, salinity, it's all in only one parameter. So we cannot choose. In this case, it's true that in case of the water, as all this happens in water, we could, this is true, we could change in the same, we would put in the model what you're saying, just by deciding that in some parts of in some times, there's no water, and so all the structures would get stuck and they would later come back. So we could do some kind of small changes, but it is definitely too abstract to be able to decide whether it's pH or it's temperature, what is really changing things. The idea is that you need something that fluctuates, and it's logical because you need a structure to grow, first to polymerize, then you need another one to gets on it and grow, and then you need to divide it, and so for this kind of to divide it and let it go to another part and be copied again. For that kind of you need an environment that lets you grow and then destroys you. This is typical fluctuating.
Thank you. Yeah, that was more or less what I was.
So I think we are out for today. Thank you so much, Jacobo. Very interesting.
Thank you all. Thank you, Rita.